everybody, I'm Tall.
And I'm Small.
And today we're doing Khan Academy's Cells section, Cell Membrane Questions. Okay, first question. Which intermolecular process primarily drives the formation of a bilayer when phospholipids are added to water? Phospholipids self-assemble into a bilayer due to the strong affinity they have for each other. A bilayer arrangement maximizes the strength of van der Waals forces among phospholipids. Lipids cause water to arrange in an ordered, unfavorable, cage-like structure. Forcing lipids into a bilayer reduces this effect. Or, the ordered arrangement of a bilayer is more favorable than the disordered state of individual free-floating phospholipids. Okay, so did you see anything that jumped out to you that you would start at?
No, I don't like the wording of any of these answers.
Yeah, I'm kind of with you on that. Okay, so let's take them one by one. So, first off, they're asking, intermolecular process drives the formation of a bilayer in water. So, phospholipids self-assemble into a bilayer due to the strong affinity they have for each other. Maybe. I mean, it's not really the affinity that they have for each other. It's the hydrophobicity.
Yeah, it's the need to exclude water and to increase the entropy of water when they form those hydrophobic groupings together.
Right.
So yeah, I don't like any of these answers.
Okay, then the second one, a bilayer arrangement maximizes the strength of van der Waals forces among phospholipids. I don't think that's why they come together. Lipids cause water to arrange in an ordered, unfavorable, cage-like structure. Forcing lipids into bilayer reduces this effect. That doesn't sound right at all. The ordered arrangement of a bilayer is more favorable than the disordered arrangement of individual free-floating phospholipids. Is more favorable. I kind of like that answer. The ordered arrangement of a bilayer is more favorable than the disordered state of free-floating phospholipids.
That's what I went with, and it's wrong.
Oh, no, that's wrong.
Yeah.
What was the correct answer? Wow.
Yeah, I hate that.
Okay, so lipids cause water to arrange in an ordered, unfavorable, cage-like structure. Forcing lipids into a bilayer reduces this effect. What do you think?
I don't like the wording at all because we don't force lipids. You know, they assemble that way because it is more favorable.
Okay, so it says in the hints, notice two answers are essentially the same. Phospholipids self-assemble into a bilayer due to the strong affinity they have for each other, and a bilayer arrangement maximizes the strength of van der Waals forces among phospholipids. Typically, if two answers are the same, so strong affinity they have for each other, van der Waals forces. Okay, yeah, that would make sense. So that could have eliminated both of those. So their explanation for the answer is the formation of a phospholipid layer is primarily driven by an increase in entropy. Okay.
But they didn't write that as an answer choice.
Well, but you knew it.
Yeah.
You mentioned it. So, driven by an increase in entropy surrounding water molecules, lipids cause water to arrange in an ordered, unfavorable cage-like structure called a clathrate cage.
Okay. Forcing
lipid into a bilayer reduces this effect. So it's because when you have individual free-floating phospholipids, it creates a higher entropy in water?
Free floating?
Yeah.
Lipids, no. It would decrease the amount of free positions that water can be in, therefore decreasing entropy, decreasing the freedom of water.
Because it puts it in the cage structure?
Yeah, but I've never heard of that cage before, so that's something I just truly didn't know.
Yeah, I mean, I guess that answer makes sense from an entropy standpoint, but we didn't know if they were just like throwing some like made up stuff at us.
Yeah. Okay,
but that's good to know. It's a very basic principle that you probably will be tested on the MCAT. So it's good to know that the water does actually start forming ordered structures around phospholipids, which decreases entropy.
Yep.
So phospholipids assemble together. Interesting. Okay, next question. Which statement best describes how cholesterol affects cell membrane fluidity? Cholesterol decreases fluidity at high temperatures and decreases fluidity at low temperatures. Cholesterol decreases fluidity at high temperatures and increases fluidity at low temperatures. Cholesterol increases fluidity at high temperatures and increases fluidity at low temperatures. Or cholesterol increases fluidity at high temperatures and decreases fluidity at low temperatures. So, I know now that cholesterol does actually function at high and low temperatures. I guess I forgot that. So when you have low temperatures, I think you add cholesterol to make the membrane more fluid.
Yeah, and some cis unsaturated tails.
Right. So if we have low temperatures, so the second half, we would increase fluidity. So that narrows it down to two choices. So we have to find out at high temperatures, does it increase or decrease fluidity? So I would guess, because at high temperatures, you become more fluid. So I guess if you add cholesterol, would it somehow decrease it?
Yeah, so the interesting thing about cholesterol is that it increases the range at which you can remain fluid. And what that means at the top half of that range is that it just keeps the membrane together. So you're right, it would decrease fluidity. Because what happens is it's so bulky that at low temperatures, like you said, it works to allow those tails from sitting right on top of each other. And so the membrane will remain fluid. But at really high temperatures, those lipids, if they get too high, are going to have a tendency to not fly apart, but come apart, right? So the big, bulky cholesterol sits in the middle of those tails, and they only have so much wiggle room. They can still wiggle because there's more kinetic energy at higher temperatures, but they're stuck in the membrane because of cholesterol.
Okay, yeah. So it increases so cholesterol just increases the full temperature range that a membrane can exist at.
Yeah, exactly. So
Okay.
I almost put, or actually I think I did put the first time that it increases it at both high temperatures and low temperatures, but it just increases the range. And so its function at the high end and the low end are kind of opposite.
Okay, so it would decrease fluidity at high temperatures.
Yep.
And increase fluidity at low temperatures. And that's the correct answer. Okay, next question. Compared to a typical animal cell, the cell membranes on the paw of a polar bear would most likely have an increased concentration of which macromolecule? Okay, so compared to a typical animal cell. Saturated phospholipids, potassium channel proteins, unsaturated phospholipids, aquaporin proteins. I think we could rule out aquaporin and potassium channels.
Yeah.
Because if you're talking about a polar bear's paw, you just want... Well, it's going to be cold. They said a polar bear specifically.
Yep.
That's cold. So if you're cold, you want unsaturated phospholipids because they are more fluid.
Yeah. Are you ready for an inappropriate way to remember this?
Please.
When things get too cold in the bedroom, you need to add some kink. You need to add some unsaturated phospholipid tails.
When things get too cold in the bedroom, you need to add some kink.
Yep.
People, you all better remember that on the MCAT.
And so we see this a lot with a bunch of fish species. During the day, they can have saturated fatty acids in their membranes, but then towards nighttime, when the temperatures drop even lower, those conformations of the lipid tails actually become unsaturated.
So, and that's also why when you get fish oils, like when you're buying fish oil supplements, it's always like cold water, Alaskan salmon, something like that, because it's really cold and they have more potentially unsaturated phospholipids in their membranes.
Yeah, I didn't know that.
Oh, well, yeah. I was kind of just guessing.
Oh, well, yeah, I'm sure that if that is like what you typically see in supplements, that would be a really good bet for why.
Okay, very cool.
And trans is like not natural, right? It doesn't happen in nature.
Right. Okay, next question. Which molecule diffuses through a membrane most quickly? Benzene, ethylene, glucose, urea. Benzene is non-polar. It's pretty small. But ethylene is also non-polar, I think, and smaller. Also, benzene is aromatic, so I don't know about that. Glucose has a bunch of stuff going on, and I'm pretty sure we have glucose transporters specifically for this reason. Urea is charged, if I'm not mistaken.
Urea is not charged. It's just a carbonyl with two amino groups on either side.
So I'm between ethylene and urea. I guess I would go with ethylene.
Yes, and that is the right answer. So it's small, non-polar, and is able to traverse the membrane without like a channel or a pore that's specific for it.
Okay, so urea, if it's you said it's a carbonyl with two amino groups. So is the issue that it's polar or
Yeah.
that? Okay, yeah.
Yeah, it's definitely polar.
Okay. And then glucose is like, I guess, kind of polar and big.
Yep.
And then benzene is big.
Mhm.
Okay, so ethylene, yeah. Think about the small, yeah, going through the membrane, you want small and you want nonpolar. Okay, next question. Which fatty acid tail would contribute most to the stability of the cell membrane in a thermophilic bacterium? Okay, so they show us the pictures of the fatty acids, but what we're looking at here is thermophilic bacterium, which means they're really high temperatures, which means we need a fatty acid to increase fluidity.
Well done.
So the first one we see here, I think that's a trans bond.
Mm-hmm. And we know
that trans fats don't exist in nature.
Yep.
And they are actually very rigid.
Mm-hmm.
So that would be the opposite effect, the opposite desired effect. Ooh, don't we also want... So it's not just unsaturation. There's a length thing, too.
There is.
Shorter is more fluid?
Yes, because longer fatty acid tails are able to stick on top of each other, and there's more force because there's more carbons that are right next to each other. So there's a higher overall van der Waals force holding those two together.
Okay, so that's where van der Waals force... I remembered it had something to do with inside the membrane.
Yep.
So it's holding what do you call it? Reduced, saturated fatty acid chains. So if we have a thermophilic bacterium, we have a trans bond. That's not going to be it. We have the second one, which is the same length, but it's a single cis bond, a mono unsaturated fatty acid. Then we have a very long, fully saturated fatty acid. And then we have a saturated fatty acid that's the same length as our unsaturated one. So I'm thinking that if this is a thermophilic bacterium and the membrane is, I might have said it wrong at the beginning. If this is thermophilic and it's really, really hot, we don't want a fluid phospholipid. We want a really rigid one. But I don't think we want trans fats, just because I don't know why you wouldn't want a trans fat. I guess I don't know enough about trans fats. I just know that they're bad because the government tells me so.
Yeah.
So I would go with the really long, fully saturated one.
It's because it asked us for stability.
Yes, and that is the correct answer. So yeah, remember that the longer a fatty acid is, the more van der Waals, like you said, more van der Waals forces you're going to have, and the more saturated it is, once again, it's going to be more rigid. It's not going to have those points to bend at. Okay, next question. Glucose typically enters the cell through which mechanism? Simple diffusion through the cell membrane, facilitated diffusion through a carrier protein, pinocytosis through a channel protein, or active transport by a glucose transport protein? I don't think it requires energy. I think it's facilitated diffusion through a carrier protein.
Doesn't it come in with sodium?
Yes. Oh, no, that totally happens. Sodium follows its concentration gradient and takes glucose in, I think. I'm going to click that. Yay, it was right. But let's look at those hints just to be sure. Okay, glucose is too large and polar to move quickly through the cell membrane by simple diffusion. Typically, the concentration of glucose outside the cell is higher than inside the cell, so concentration gradient. Since glucose is moving down a concentration gradient by entering the cell, the process does not require ATP. Okay, so that's the real kicker for ATP, is concentration gradient.
Yep.
Glucose typically enters cells by facilitated diffusion through a carrier protein. And I think you're right that it does couple with sodium.
Yeah, and I think it's a symporter, so not only are you going down your concentration gradient for glucose, you're doing the same with sodium, and it makes it even more favorable.
So maybe that's where, I wonder if there's any kind of connection to like sodium potassium ATPases, like pumping sodium back out because you need that concentration gradient to like, pair up with glucose and go down.
Yeah, definitely.
Yeah, I wonder.
It all feeds together.
That's what I've learned about metabolism. Okay, next question. Which statement represents a notable difference between simple diffusion and facilitated diffusion? So, unlike simple diffusion, facilitated diffusion can occur in any type of cell. Unlike simple diffusion, facilitated diffusion can transport ligands against a concentration gradient. Unlike simple diffusion, the rate of facilitated diffusion is limited by the number of transport proteins in the membrane, or, unlike simple diffusion, facilitated diffusion requires energy in the form of ATP. So if they say the word diffusion, there's two things. One, it doesn't require energy.
Yep.
And two, diffusion means a concentration gradient, which I guess is kind of the definition of not needing energy.
Yep.
Based on our previous question. So I would say ATP is right out. Limited by the number of transport proteins in the membrane. Well, I assume that would be the same with simple diffusion or facilitated diffusion.
Well, do you remember the definition of facilitated diffusion?
It's just diffusion that requires some kind of protein to mediate the passage of whatever the thing is.
So read that answer choice again.
Unlike simple diffusion, the rate of facilitated diffusion is limited by the number of transport proteins in the membrane. Oh my gosh. I'd like to think I would have come back and saw that, but I might not have.
Yeah.
Okay, let's read the other answers just to see if I would have. Okay, so unlike simple diffusion, facilitated diffusion can transport ligands across a concentration gradient. No, because if they're both diffusion, that's what they're doing. And then unlike simple diffusion, facilitated diffusion can occur in any type of cell. Now I think simple diffusion could as well. So yeah, okay. That was tricky. I don't know if that was tricky for everyone else, but that tricked me a little bit. So the correct answer would be that unlike simple diffusion, the rate of facilitated diffusion is limited by the number of transport proteins in the cell membrane. Very cool. All right, we got it right. Next question, question number 8. Which type of membrane protein is most likely to be a hormone? A lipid-bound protein, a peripheral protein, an integral protein, or a steroid? So steroid isn't a protein.
Yeah.
A lipid-bound protein, I don't know. Peripheral protein? That means it's just on the outside of the cell.
Yep.
Integral protein, does that mean that it's like a transmembrane, or does that mean that it's only in the middle?
No, it does mean transmembrane. And so lipid bound is like a little lipid tail on it. And so it just like sticks in and is anchored there, literally a lipid anchor.
Oh, okay.
So lipid bound to me and integral both denote that they're, staying put. But peripheral, I guess, is the only one that kind of makes sense. I don't know. I'm thinking about GPCRs and those G proteins moving. Because they are peripheral proteins, they can move along that membrane on the inside.
Well, it also says what type of membrane protein is most likely to be a hormone.
Yeah, I don't know what they're asking.
I mean, it can't be steroid. Steroid is not a protein.
Right.
Wow, I really wouldn't even know how to answer this. I'm thinking peripheral protein.
I mean, the other two are definitely bound and stationary.
Yeah, bound and stationary. All right, 1, 2, 3. Oh, we got it.
Okay.
Let's look at those hints. Okay, hormones move through the circulatory system, acting as signaling molecules to specific organs. A steroid is not a membrane protein. We got that far. An integral protein cannot easily leave the cell membrane and would therefore be a poor signaling molecule. Lipid-bound proteins remain within the phospholipid bilayer by the fatty acid tails of phospholipids. They would also be poor signaling molecules. And then peptide hormones like insulin and growth hormone interact with integral membrane protein receptors in cell membranes. These hormones can be considered peripheral membrane proteins. Okay, that makes sense.
Yeah.
All right, next question. The rate of osmosis across a cell membrane depends upon which of the following? Is it... okay, so they give us a list of possible answer choices. Intracellular solute concentration, extracellular solute concentration, polarity of solutes, molecular weight of solutes, or the presence of aquaporins. And the actual answer choices are just combinations of those. So we need to find out which of these will affect the rate of osmosis. So the movement of water following salt. That's osmosis?
Yeah.
Basically.
Solutes in general.
Okay, yeah. So intracellular solute concentration, I would assume.
Yep.
Extracellular, yeah.
It's our hypertonic, hypotonic, isotonic.
Yep. So it can move either way. The polarity of the solutes, I don't think so.
They're not moving.
Right. Molecular weight of solutes, I also don't think so.
Yep.
The presence of aquaporins, yeah, because that's how water moves across. It's facilitated diffusion.
Nice.
Which, if you didn't know this, if there weren't a lot of aquaporins, that would limit the rate of diffusion.
Oh, you think?
So we're going with 1, 2, and 5, which is the first answer choice. And it's correct. Very cool. Last question. In nerve cells, sodium-potassium pumps exchange 2 potassiums for 3 sodiums across the cell membrane. What is the primary purpose of this exchange? Is it to propagate an action potential, to store electrical and chemical potential energy, to increase the concentration of sodium inside the cell, or to increase the concentration of potassium outside the cell? So it is potassium goes in and sodium goes out through the enzyme?
Yeah, the sodium potassium ATPase pump is an enzyme. And I remember this by remembering pumpkin. Pump K in.
Oh,
and
pumpkin.
you pump 2K in and 3Na out because you have an overall negative charge on the inside of the cell.
Okay, so this would immediately make the last 2 answer choices just rubbish because you're not trying to increase the concentration of potassium outside the cell if you're trying to pump K in.
Yep.
So that would also mean that the converse of the converse of that, which is the other answer choice, increase the concentration of sodium inside the cell, is the opposite of what we're doing.
Yep.
You don't propagate an action potential with the sodium potassium ATPase. In fact, leading to the final answer, you actually are trying to store electrical and chemical potential energy so that in the case of an action potential, you can propagate one in the future. But it itself does not. It just sets up the environment.
Perfect. And then after we receive the signal, we depolarize by letting in a whole bunch of sodium. And then what do we do to the sodium channels in order to repolarize? Do you remember?
So sodium channels get deactivated then inactivated, or inactivated then closed?
Yeah, it's weird. Yeah.
I should know this because I study them. I only look at the structure.
It's our dry lab expert. There we go.
Yeah.
So they're wide open when we're depolarizing, and sodium is flooding into the cell, and we're getting really positive, and then we inactivate, we close, but not only do we close, we also inactivate with a ligand. And that is so we can ensure a unidirectional signal. So that's why your neuron doesn't fire in both directions.
Okay.
Is that not awesome?
Okay. That makes a lot of sense.
Yeah, and so when those are inactivated, in order to repolarize the cell, we start opening some leaky potassium channels and letting potassium flood back out.
Right. Okay, yeah, that repolarizes, hyperpolarizes, and then brings you back to normal.
Exactly.
All right, very cool. Strong work, gang.